Hello everyone, this is our bacterial structure lecture second. Now in this lecture we are going to understand structure external to the cell wall. Let's start our lecture. Now see, this is bacterial structure. We have learned the different body parts. Now first we are going to understand the structure known as flagella. Now see, the flagella, the bacteria can be motile or non-motile. The motile forms swim by means of small flexible whip-like appendages called the flagella. Now flagella is what? It is a small flexible whip-like appendages which is coming from the cell membrane. They are much thinner than the flagella or cilia of eukaryotes. A typical bacterium flagellum measures about 120 angstrom thick and 4 to 5 inch long. Chemically, they are made of a protein known as flagellin protein, and the molecular weight is around 40,000. The bacterial flagellum do not contain any 9 plus 2 arrangement as like eukaryotes. The lack of flagella known as atrichous, for example, Diptheria bacillus and many cocci, example, Lactobacillus and Pasturella. Now, these are the some types of flagella where the monotrichous flagella means the only one flagella present at the one end of bacterial cell known as monotrichous. Example is called Ravibri. Second is a Lophotrichous means a bunch of flagella which presents at the only one end known as Lophotrichous flagella. Example Spirillum iridum. Amhitrichous flagella means the bunch of flagella presents at the two ends or the both ends of the bacteria known as Amhitrichous, example Spirillum and Nitrosomonas. And last one, Peritrichous means the flagella present all over the body known as Peritrichous flagella, example Salmonella and. So bacterial flagella mainly consists three parts, that is the filament and the hook and the basal body part. Filament, hook and basal body part. Let's see filament. Now filament is the hair-like structure which is concerned to the hook. There is a wide region at the base of the flagellum called the hook. Hook penetrates into the cell wall. Hook penetrates into the cell wall and connects the filament to the motor portion of the flagellum. This is the motor portion of the flagellum. The basal body is anchored into the plasma membrane. Now see, this is the basal body and this basal body is present in the plasma membrane. There are presence of a series of rings like L series. This is L series, which is present in the lipopolysaccharide. That's what L. This is a P series. This is present in the peptidoglycan. That is a P, and then MS ring, and there is a C ring, which is the down to the MS ring. Okay, M, S, and the C are surrounded by a pair of proteins called the MOT. Now, this MS ring and the C ring is the surrounded by a protein known as MOT protein. These proteins are actually drive the flagella motor, causing the rotation of the filament. Now, this MOT protein are helpful. Proteins are helpful for the like what rotation of the filament. Another set of proteins called the flea proteins, which are helpful in the motor switch and the motor like switch off and switch on. Okay, now what is the function of flagella? The main function of flagella is the movement of bacterial cell through the liquid medium. Sometimes the bacterial flagella are used to sense the unfavorable environment, such as changes of temperature, pH, or chemicals. They are also useful in the movements called taxus. Chemotaxis is a response to the chemicals. Phototaxis is a response to the light. So this is all about the flagellum. Now next part is the capsule. It is also called as slime layers, or it is also called as a glycocalyx. Now it is a viscous layer which is the outermost layer of the cell wall, and it is a gelatinous sheath covering around the bacterial cell. If the covering is so much loose, it is known as slime layer. It is also called as capsule when it is relatively narrow and well defined. They consist two types, macrocapsules and microcapsules. Now see, the macrocapsules are 0.2 micrometer thick. They can be seen under light microscope, while the microcapsules are less than 0.2 micrometer and they cannot be seen under the light microscope. The chemical composition of capsules, they are totally made from polysaccharides or the polypeptides. In Acetobacterium hylinum, the polysaccharide is mainly present cellulose, and it is a homopolysaccharide, and it is made from the glucose units linked to beta 1,4 linkage. Pseudomonas aeruginosa secretes the heteropolysaccharide consisting D-glucose, D-galactose, D-mannose, D-glucuronic acid and the D-rhamnose residues. What are the functions of capsule? So main function of the capsule is to protect the bacterial cell from the outside environment. They also protect the pathogenic bacteria from the phagocytosis and also serve as a storage product. Certain pathogenic bacteria owe their virulence to the capsule polysaccharide or slime. Sometimes they protect bacterial cell from the desiccation. Next part is the mesosome. It is a complex localized infoldings of the cell membrane. They found in gram-positive and gram-negative bacterial cells, particularly well developed in bacilli only. The mesosome consists of an invegetated cell membrane with the many vesicles, tubules, or laminal walls filling the invagination. According to Salt and Owen in 1976, they have suggested that mesosomes may arise by tubules of the outer half of the lipid bilayer. Now, this is the structure of the mesosome, which is the smallest part or inward part of cell membrane inside the cell. For example, see, this is cell membrane. Okay, this is a cell membrane, and from this cell membrane one part is coming inward or inside the cell membrane or inside the cytoplasm, known as mesosome. And if you see the mesosome, now see, this mesosome contains mainly tubules and the mesosomal sac. Okay, next point is the, according to Huggins in 1974, the tubular have the artifacts resulting from fixation, centrifugation, and the filtration. What are the functions? Mesosome initiate the DNA replication and septum formation during the cell division. They are useful in respiratory activity. They are useful in the electron transport chain. So this is all about the mesosome. Next part is pili. It is also called as fimri. Many gram-negative bacterial cells contain a hair-like projection known as the fimri or pili given off from their surfaces. In the gram-positive bacteria, similar structures have been observed in the Corynebacterium renale. Their length varies from the 0.2 to 20 micrometer and their width varies from 30 angstrom to the 140 angstrom. Classification: according to Otto 1975, they have divided into 5 groups. First one, group first. It works as adhesive organelles determined by the chromosomes, divided into 4 subtypes. Group second includes 6 pili, that is F pili, present like 1 to 10 per organism. It is longer and thicker and in with the terminal knobs. Group third is thick hollow tubes and it is around 3 micrometer long and 400 to 600 angstrom wide. Group 4, it is flexible rod-like found in Vibrio Pseudomonas, promote bacterial motion. Group 5 is a poorly arranged contracted tubules found in Rhizobium lupini and promotes conjugation. Group 6 is found in Corynebacterium renale. Diameter is around 25 to 30 angstrom. It acts as antigens. Now, what are the functions? Fimri helps to cells adhere to the surface. They are involved in the forming biofilms. Pili are involved in the adhesion during conjugation, such as pili are called as sex pili. Next is sheaths. Now see, some species of bacteria of fresh water and marine environment form chains or trichomes which are enclosed by hollow tube called a sheath. Sheaths may be sometimes impregnate with the ferric and manganese hydroxides which strengthen them. Next part is the prosthesia and stalks. These are semi-rigid extension of the cell wall cytoplasmic membrane. They are characteristic for a number of aerobic bacteria from freshwater and the marine environment. These processes may be single or may be several. The main function of processes is that they increase the surface area of the cells for nutrient absorption in the diluent environment. Stalks are non-living ribbon-like structure that are excreted by the cell. The stalk aid in attachment of the cell surface, for example, parectomyces. This is all about the some body parts of the bacterial cell. Thanks a lot.